Hello everyone, this is Richard from Modern Healthspan. In the recent ITP paper that presented the results of Astaxanthin extending lifespan, they also talked about Fisetin, which did not extend lifespan. As this molecule has been shown to extend lifespan and healthspan through its senolytic effects in previous studies, I wanted to have a look at the results in a little bit more depth. In the recent paper, Astaxanthin and meclizine showed significant increases in median lifespan by 12 and 8% respectively. We talked about the results for Astaxanthin in another video, which is linked above. In the set of tests, the ITP also looked at 5 other interventions, one of which was Fisetin. None of these, including Fisetin, increased lifespan by a significant amount with the schedule and doses used in the trial. A very quick review of the ITP as a little background. This is the interventions testing program, which is part of the NIH. They use genetically heterogeneous mouse model rather than the genetically identical models used in most other studies, and they test the intervention's effect on the lifespan of mice. They run the tests in 3 locations at the same time: University of Texas San Antonio, University of Michigan, and the Jackson Lab. Why was Fisetin included in the ITP? Senescent cells increase with age and have been shown in trials to be related to some of the diseases of aging. Removing them with a senolytic, such as Fisetin, has been shown to help mediate the effects of the diseases. They then refer to a set of trials which have had positive results with Fisetin, particularly the paper by Dr. Yousefzadeh, which showed a reduction in markers of senescent cells, and at a dose of 500 parts per million, extended lifespan by 27 to 30 months. The ITP based their dosage of 600 parts per million on that used in the previous trials. Generally, young mice are able to remove excess senescent cells naturally. They therefore chose to start the treatment on 20-month-old mice, about equivalent to a 60-year-old human, when senescent cells would be expected to have started accumulating. As senescent cells take some time to form, possibly 4 to 6 weeks in the human, it is possible that a continuous regimen of senolytics is not needed, and instead the drug can be taken intermittently. Hence the trial, they tried 2 different protocols, one with a continuous dose and one for 3 days every 2 weeks. Let's have a look at what the dose was they used and how this compares to the doses used in human trials. Parts per million is equivalent to milligrams of drug per kilogram of chow. Mice eat 10 to 15 grams of food per 100 grams of body weight, so I will use 12 grams. 20-month-old mice weigh about 30 grams. Note that in this calculation, the ITP use mice of both sexes and a different strain than the one that I used to estimate the weight from. Female mice weigh less than males. For the calculation purposes, I have taken the value of 30 grams, which would mean they would eat 3.6 grams per day. The dose was 600 parts per million. So based on how much chow the mouse would eat, this comes to 2.16 milligrams. The mice weigh 30 grams, so this comes to 72 milligrams per kilogram of body weight. The standard conversion factor is 12.3, so for a human, this would be 5.8 milligrams per kilogram. And for a 75 kilogram person, this would be approximately 438 milligrams. A couple of comments to add here. The Mayo Clinic is currently running some human trials, and the dose that they are using is 20 milligrams per kilogram for 2 days in a row, either once every 2 weeks or once a month. This is based on the principle of removing senescent cells, which then take some weeks to reform, so the dose can be taken intermittently. My understanding of this dose is that it is more than you would want to take every day, but it is okay on an intermittent basis. In the ITP trial, the dose for daily and intermittent was the same. Here are the results for the Fisetin life extension trials. These graphs show the Kaplan-Meier survival graphs for the pooled male and female cohorts from all 3 locations for 4 of the interventions. which include the cycled and continuous Fisetin. And as we can see, neither had a significant effect on either median or total lifespan. They also looked for the presence of senescent cells in a subset of the mice in liver, kidney, and brain tissue by checking the expression of P16, a marker of senescence. For all tissues, there was a significant increase in senescent cells between the young and old controls, which is expected and shows that the older mice had accumulated senescent cells. However, there was no significant difference between the old controls and those on either of the Fisetin regimens, implying that Fisetin had not significantly reduced the senescent cell burden in the mice. Let's have a quick look at the previous study. Fisetin is a therapeutic that extends health and lifespan, from Dr. Youssef Zadeh. The first graph is showing the results of luminescence in mice treated with P16 luciferase reporter. We can see that the Fisetin-treated mice had much lower levels of P16. And on the right hand, that the Fisetin mice had a longer median and absolute lifespan. The ITP team did not think the difference in the results was because of the dosing or regimen, though they did use a much larger cohort in the tests and so had much more power. They proposed some possible reasons, such as different mouse stock or ways that the mice were kept, and suggest further studies. Looking at other mouse studies, they mostly use 100 milligrams per kilogram, which is close to my calculation for what was used in the ITP. In humans, there is no recommended dose, but let's call it a typical dose for daily usage is 100 milligrams, while that for intermittent usage is 20 milligrams per kilogram. For me, as a 66 kilogram person, that would be around 1.3 grams, 13 times as much as the daily. In the mouse trials, the intermittent and the daily were the same. This does not explain why there were different results in the two studies, but it would make sense to have a higher dose in the intermittent group, as judging from the lack of impact on P16 on the dose, they were unable to clear the senescent cells. As the Fisetin did not clear the senescent cells, it would be really good if they could try a higher dose of Fisetin in an intermittent fashion, or perhaps some other senolytic to see how a senolytic would impact lifespan. Thank you for your attention, and I wish you all well.